right? Yep. All right, welcome back, everyone. I would like to welcome you to our next talk by David Galevski.
Galevski.
Okay.
Whatever.
Sorry. All right. And he will be talking about how to improve the diagnosis of genetic disorders using machine learning. Enjoy the talk, everyone, and have fun.
Okay, hi everyone. My name is David, and today I'll be talking about using machine learning to improve the diagnosis of genetic disorders, which is basically the work that we do at Netcetera together with a Danish startup called gMendel. So basically the story so far goes like this. The diagnosis of a rare genetic disorder is kind of a long and painful journey that sometimes can take up to 8 or more years to diagnose. And the timeline goes something like this. So the child is born, it shows some first symptoms, and then you go to the doctor and they don't know what the problem is, or they give an incorrect diagnosis. So you go to a specialist and then another specialist and so on and so on. And this takes a lot of time and a lot of money until in the end you decide to do genetic testing and you get test results and you find out sometimes even too late that there's a problem. So what would the solution for this look like? Well, the main idea is to develop a novel diagnostic tool that would analyze DNA in real time and provide accurate diagnosis of genetic disorders. And tests like this already exist, but the main thing it is to make it cheap and affordable for everyone. So what we're planning to do is basically there are 3 kinds of tests that you can develop. The first one is a newborn screening test, which happens here and is done after the child is born. You take DNA of a child and you screen for basically a lot of different genetic disorders. The second kind of test is the non-invasive prenatal test, which happens before the child is born. So you screen the child for any genetic disorders that might come up. And the third kind of test happens even further down, which is basically screening of the parents if they're carriers of any rare genetic disorders. So you can they can know beforehand if they even want to have a child. So today we'll be focusing mainly on this first kind of test, which is the newborn screening test, because that's what we already have a product on. The work is already done. So the main focus will be the Klinefelter syndrome, which is just one syndrome. We decided to kind of develop a whole product just for one syndrome to see if we can do it, and then kind of continue with everything else. So basically, the next steps are to either develop newborn screening tests for more disorders, and then go and develop the other tests. So the Klinefelter syndrome affects only boys, and it happens because the child is born with one extra X chromosome. So males have XY chromosomes. In this case, they have XXY chromosomes, and some of the symptoms are infertility, low testosterone, less muscle mass. They're taller and skinnier, and so on and so on. So, and the next steps would be to extend this to more syndromes. So first, let's give a short DNA crash course. I guess everyone knows this, but their DNA looks like this. There are 2 strands, and there are 4 base pairs, which are basically the letters that make up everything. And the A on one strand is always connected to a T on the other, and the G is connected to a C, and the other way around. We as humans have 23 pairs of chromosomes, 1 from each parent, and the sex chromosomes are over here. XYs are for males and XX are for females. And why are we doing this now? Well, this is basically why, because the cost of whole genome sequencing has dropped rapidly in the past 10 to 20 years. We can see it's even faster than Moore's law, the price. Right now, in 2020, it was $1,000 for sequencing of the whole genome of one person, and now it's even probably closer to $600 to $700. And our idea with the test is to not sequence the whole genome, only sequence important parts that we already know where to look for, so we can make it even cheaper for a single person to do, because we can sequence a lot of people at once. So this is basically how the whole pipeline looks like right now. The first step is the preparation of the library. This happens in Denmark in the lab. This is not what I do, but so basically they take DNA swabs and prepare everything, and then they send the DNA data to a sequencer, which gives out basically just text files of DNA, and then our work starts here, basically, with FIVEA, which is what we call the platform for detection of diseases. So the lab part basically looks something like this. You take a swab of the cheeks, basically similar to the COVID rapid tests. Then in the lab, they use something called primers to target specific regions of interest inside the DNA. And then they also add barcodes to the DNA, and the barcode is basically something that tells us this piece of DNA comes from this person. So after that, they do PCR to kind of like multiply the samples. And then let me say a little bit more about barcoding, which is very important, as we'll see later on. This barcoding part, this is how it looks like for biologists. It's kind of hard even for me to understand. It's like a two-step PCR process, and they attach something. But for software engineers, which is what I am, it basically looks like this. We have a DNA sequence, which in our case is about like 1,000 characters long. And we add a barcode at the start and at the beginning of the sequence that tells us this sequence comes from this patient. But this is bioinformatics, and everything is not as simple as it looks, so there's always noise. Usually it's in front of the barcode, sometimes it's inside the barcode, sometimes it's after, and it's very hard to kind of find where the barcode is and determine which patient this DNA comes from. And now we move on to the sequencing part, which for sequencing we use the Oxford Nanopore devices. There are 3 devices, maybe there are more now, but the MinION, the GridION, the PromethION, and they're pretty much the same. They're just one is faster than the others. And this is how the sequencing looks like. They use a nanopore protein or something to split the strands, and then they measure electrical changes or whatever, and then use machine learning on that to figure out which basically DNA base pair it is. And now after the sequencing is done, we get a text file of ATGC sequences, and we start the machine learning work. So when we get a text file, we first have to, basically it looks like this. We have to check the sequence quality of all the sequences. And together with the text file of the sequences, we also get qualities for each sequence, and we kind of throw away the ones that we don't need. Afterwards, we move on to the demultiplexing, which is the reverse of the barcoding. We now have to find the barcodes and assign them to which person they come from. There are already tools that do this, but we kind of wanted to, they were really slow in our case, and we wanted to implement something of our own, and we called it Torchlex, which despite the name is not connected to machine learning on PyTorch in any way. It was a purely algorithmic approach where we used like edit distances and other string matching algorithms to basically make a very fast algorithm for demultiplexing, which we actually have a poster presentation on it. It was like, in our case, 10 times faster than the competitive tools. So anyways, we find the persons, and we say, okay, this sequence comes from person 11, this one from person 23, and so on. And then we move on to find which region of DNA the sequence comes from. So for this, we use already known sequence alignment algorithms that are known in bioinformatics, like BLAST or Minimap2 and so on. And basically, we can say, okay, this sequence comes from this chromosome or this exact region of a chromosome. And basically, after we have all this, we have a table that looks something like this. And if we have a table, we can kind of already do some basic machine learning on it. Basically, we have, for each person, we know, okay, there are like 1,000 sequences that come from chromosome Y, 10,000 from chromosome X, and so on and so on. And kind of after we get this, there was like a very long back and forth process between our team, the data science team, and the biologists to figure out kind of like what columns to add next and what feature engineering to do. So basically, in the end, we had something like we had control samples. So like the first 8 barcodes, I think, are from people that we know are healthy and we know like what to expect from them. And then we calculate some distance metrics from them. We add some ratios and so on. And also, there's one extra step that we do that helps a lot for noise reduction, which is we call it technical replicates. Basically, 4 barcodes is 4 copies of the same person. And we do that to kind of make the whole thing more robust to noise, because there's a lot of noise. So yeah, after this, we can even kind of already plot the data with like some PCA and reduce it to 3 dimensions. And we already see that we have like a pretty easy problem, basically. It's like the healthy ones are over here and the Klinefelter samples are over here. So it's pretty much an easy problem if we only have control samples and Klinefelters. But when you add more diseases, it gets kind of harder. But again, this is reduced down to 3 dimensions. So if we train a good machine learning algorithm, it should be able to figure out which samples come from which disease. And we also did one test it's called a mosaicity test, which is basically they mixed patients with Klinefelter with healthy patients, like the DNA. And for example, they mixed 80% of Klinefelter data with 20% of healthy patients. And we should be able to see that as like 80% Klinefelter or something like that. And basically we see that here. So these ones have like 0% Klinefelter, and the more you mix it, the more they go down, which was kind of a good sign that told us, okay, we're on a good track. So the machine learning slide, here it would have been amazing to say that we've used some state-of-the-art GPT-3 or transformer-based methods or whatever, but as we can see in this case, for Klinefelter syndrome, we kind of did really good feature engineering so that we didn't have to use any of that. And another constraint was there isn't a lot of data, because we need to find a lot of patients that can have Klinefelter and other syndromes and are willing to give us data. So props to the team that found us the data that we had, but there wasn't a lot to work with. So basically we stuck just to classic machine learning. We first tried tree-based models like XGBoost, LightGBM, and so on. But somehow in the end, we stuck with support vector classifiers. And basically the final models were an ensemble of, I think, 5 support vector classifiers that were each trained on a little bit of a different data set to account for the noise and different kind of errors that can occur. And the output of the FIVEA platform is a JSON file that looks like this, which basically says, okay, person 1, the result is inconclusive. There's a 16% probability that it has Klinefelter. Person 2 has 0% probability, and so on and so forth. And this is basically the test that we did. We trained the platform with something like I think 200 patients, which was, again, not a lot. And we tested it on 40 patients, and the sensitivity and specificity are like 97 and 99%, and this is per replicate. It's like for 1 copy of the 4 that we have for a patient. So even if we aggregate the data for like all 4 copies, we can get this to like 99 plus. And we expect this to drop a little bit when we try it with more and more samples, but again, we can always add more replicates. For example, instead of 4, we can try and use 8. So the end result is that basically FIVEA 1.0.0 is certified as an in vitro diagnostic medical device. And from what I've heard, like from start to finish, it took us like 7 to 8 months, which is pretty fast in the whole certifying medical devices world. So that's pretty much it. So I'm going to give a quick recap in the end. So what we did was develop a newborn screening test, which was only for Klinefelter syndrome, which affects males, and it is the result of males having 1 extra X chromosome, so XXY instead of XY. This is the whole pipeline and how it looks like. The preparation of the library is the step that happens in the lab, so taking DNA, PCR, and so on. Then it goes to sequencing. The sequencer produces text files, which then we work with in the FIVEA platform. And we do pre-processing, which is the checking sequence quality, demultiplexing, matching where the sequences come from. Then we get the tables and we do machine learning on that, and these are the results. So that's pretty much everything from me, and thank you for your time. And if you have any questions, I guess now is the time.
Thank you very much, David. That was a very nice talk. Does anyone have any questions? Yes.
Yes, so you mentioned that you use data of 200 people.
Yes.
Was it 200 sick people or 200 in general? So was it 400 if it's equal? Or?
No, it was a bit imbalanced. I think it was, in the end, it was like 250 people overall. And it was like we had around 10 different disorders because we had to have more disorders while testing to see if there's any like misclassifications. And I think it was like 20-something samples per disorder. Okay,
because I'm really wondering here that because there are like a lot of different types of people around the world, right? How is the 200 people enough to check for every race and every different?
Yeah, I completely agree with you on that. It's a very small sample size to be able to say like confidently that you can do it. But the thing is that for Klinefelter, which is a chromosomal aberration, you just have to measure the like distribution change between chromosome X and chromosome Y, and it shouldn't account for like different races. Races shouldn't have that problem. And yes, there should be more samples, but we worked with what we got, and we are extending this as we're getting more and more samples.
Okay, I see. Thank you.
If I understood correctly, the barcode is also like a possible DNA sequence, right?
Yeah, the barcode is a DNA sequence.
So I was wondering how you ensure that the barcode is not part of anybody's DNA, like.
That's kind of taken into account in the process of creating the barcodes. The barcodes are basically human-made, and we have reference genomes, and we kind of know which kind of barcodes to generate. And they're always at the beginning and the end of the sequence, so we're not looking in the middle, and we kind of have a general ballpark of where to look at, and we know that there is no similar DNA there. Because there are reference genomes that like basically 99.9% of all DNA of humans is the same, and we have all that sequenced, and we can look to see if that kind of barcode appears in the places that we're looking at. And if it's similar, we just find another barcode that's better.
Thanks.
I hope that answers it.
So you mentioned that there was like back and forth between the biologists and the machine learning people.
Yes.
So I was wondering in general, like how much, to work on this kind of application, how much biology knowledge does a machine learning engineer or data scientist need?
Well, I didn't have much biology knowledge when I started, but I guess more is always better. But if you have a good team of biologists and people there to help you, you don't need much. You just kind of learn along the way. And now I'd say I'm pretty confident that I have like a pretty solid knowledge of like how everything works, at least in regards to like sequencing DNA and so on.
There was one question up here. Yes.
Thanks for the talk. I was wondering if you know why they decided to do newborn testing first instead of the other two options.
Because it was easier. In the beginning, we decided, as I said, to make like a minimum viable product to see what we can develop the fastest. And we did some experiments with prenatal testing, where you basically draw blood from the mother, and like 20% of the DNA inside the mother's blood is from the baby, but it's really hard to distinguish which comes from where. And the fastest thing that we could develop was the postnatal testing, which is testing of newborns. So that's why we decided on that.
Okay, thank you. Maybe can I do a follow-up? Actually, not a follow-up, a different question. How is that the product received by clinical professionals? Like, is it well received?
Well, I'm not the guy to ask for that. That's like, there are other people in the team responsible for that. But basically, the main idea with releasing this product was to show people that we can make a product and license it. so that we can like have support to make the more complicated products, basically to extend this for more diseases and then to move on from like prenatal testing.
Thanks.
So this was basically just like, look, we can do this.
So how exactly do you want to extend this model to other diseases? Because as I understand it, it's just for duplicate chromosomes, right?
Yes. Well, we've looked at, I think, 6 chromosomal aberrations, like genetic disorders that kind of are very similar to this, are duplications or missing of a chromosome, like Down syndrome and so on. And for those, we can already do something very similar to this. And for others, like SMA, which is spinal muscular atrophy, that happens because of a mutation in a gene called SMN1 and SMN2. And for that, we need a more like text-based approach, which is where we're trying other models like transformer-based methods to actually look at like the DNA sequences. Because here we just match it to a region, and then we just look at the distributions. But for other diseases, we're trying out different things.
So you're also considering like which region on the chromosome it's mapping to?
Yes.
Okay.
Yes, for this. And for example, the case that I said, the spinal muscular atrophy, there we sequence and we look at individual point mutations. Point mutation is basically like a single character mutation. For example, an A changes to C or something. And if an A changes to C at a specific place, you get spinal muscular atrophy. So for that, you need kind of a different approach.
Any more questions? Because if you don't, then there's nothing left to do for me. But thank you again, David. We have a little something for you. Nice mug with some edibles inside. And yes, thank you very much for the nice talk. And you enjoy the rest of VIScon.